I'd like to introduce myself very briefly before starting. So my name is Herman van der Putten. I joined the NCL Foundation in 2014 as research head. And after I retired from Novartis Pharmaceutical in Basel, where I was executive director neuroscience and worked for this company for the last 26 years. The title of my presentation today is Understanding the Fundamentals of CLN3 Juvenile Batten Disease and Finding New Ways to Treat. As most of you might know, juvenile Batten disease is caused by mutations in the CLN3 gene, which encodes a lysosomal transmembrane protein. And CLN3 is one out of 13 genes that are classified as neuronal ceroid lipofuscinoses, which is a small subgroup of lysosomal storage diseases that share interesting histopathological and also clinical hallmarks, despite the wide diversity of proteins these genes encode. And so one of the questions, I think, is what do they actually have in common? Well, they have in common the name, and I explained to you in subsequent slide why they have this common name. But the one thing they do have in common is that they all disrupt endolysosomal function. So mutations in all these 13 NCL genes, they all disrupt endolysosomal function. I'm not going into detail into these functions precisely, and I will focus today mainly on CLN3. But one of the key questions, of course, is, and I'll come back to that one, is are there any common pathways that are affected by all these different NCL genes? Now, I've told you that the NCLs are classified as NCL because they do share an important set of clinical features. So one of the key features is loss of vision, for example, occurs in almost all forms. The loss of motor and cognitive function is seen, and that is also why the NCLs are actually altogether are a major cause of childhood dementia. Other clinical phenotypes that occur over time is epilepsy. All these patients die prematurely. We observe in many samples neuroinflammation in the brain. The onset of the disease can vary, depends on the underlying gene defect, and it can occur at various times, either even late prenatal or early postnatal, all the way up to adulthood. Then again, this is, I think, one of the reasons why they were named like that, because they share important histopathological features. So the name comes from the deposition of lipofuscin, which is an autofluorescent material that you can observe in many cells in these patients. So this is not specific for neurons. And one characteristic of these lipofuscin deposits is that they contain some prominent proteins. In most NCL cases, this is the subunit C of the mitochondrial ATPase. And in some rarer forms, it's a lysosome protein called saposin. So these are an important part of the lipofuscin material. What all these diseases have in common is neuronal loss, brain atrophy. CLN3, so the juvenile form, is quite unique. It is characterized by vacuolated lymphocytes. And recently, there has been a very interesting study from a lab at Yale, the Chandra lab. They have compared the composition of the lipofuscin in normal aging. So they took aged mice and compared it to one of the NCL models, in this case, the CLN1 model. And what is interesting, they've done an extensive proteome, palmitome, lipidome study. And what they find is that there is actually a great overlap in the contents in this lipofuscin. So the old theory of that age-related lipofuscin and this NCL-specific lipofuscin are grossly different is probably not longer holding up. And as more of these NCLs are being analyzed, I think we might see more things, more similarities popping up. Now, I want to spend 2-3 words briefly on the NCL Foundation. we were established in 2002 after the founder found out that his son was suffering from CLN3 disease. And since then, our mission has been to promote networking, education, science, and fund and explore fundamentals of CLN3 disease, with the goal, of course, to identify and translate disease-transforming therapies to the clinic. Now, to do this, we are relying on a lot of co-funders that help us to collect money for what we think are important projects. And this is just a subset of them. I wanted to briefly show these to you. And another thing is that this is sort of a short output slide, a snapshot of recent projects we have been funding over the last 5 or 6 years, and resulting publications from these various projects that we funded. Going back to the NCLs, this is a group of diseases that occurs worldwide. The prevalences differ depending on country. You can have one form more prevalent than another, but worldwide overall, CLN3 is the most prevalent disease still. And the typical clinical course of the CLN3 Juvenile Batten disease is shown here schematically. It starts at school age. First clinical symptoms appear with a rapid loss of vision. And then other symptoms start kicking in as time progresses. And in the end, most patients are bedridden, and it ends with a premature death, nowadays usually in their 20s. Now, this is a scheme. The reality looks slightly different, because if you look at individual patients, the course and which symptom kicks in can vary greatly. So CLN3 is unique in a certain way because over 80% of the patients carry the same allele, the same mutation, which is a deletion of 2 exons. Nonetheless, if you look at individual patients, the symptoms as they kick in, apart from vision, all the others can be very different in terms of onset. So vision, it's like a clock. It starts when the patients reach the age of 5, 6, 7. It kicks in and it rapidly progresses. But all the other symptoms, and that's shown here on the left side, in red, you have a patient that has a so-called more severe phenotype, and in blue, one with a protected phenotype. And these are patients that carry exact same mutation. So this already tells you that this disease, you want to do clinical trials, is actually quite a difficult disease to work with, because unless you have very good biomarkers, it will be very lengthy and costly. I won't get into detail into these middle ones right now, but just to give you a comparison with another NCL population, the CLN2 patients, the only patient group for which a therapy is available nowadays, which is an enzyme replacement therapy. Here you see the homogeneous course of the disease as it progresses in these patients, as compared to CLN3 patients. Now back to the lysosome. As I said, CLN3 gene is a lysosome transmembrane protein, encodes a lysosome transmembrane protein. And of course, defects in lysosome metabolism are common to a large variety of diseases. And besides the lysosomal storage diseases. And it's well known that the heterozygous loss of function of lysosomal storage diseases can increase the risk for adult onset neurodegenerative diseases. And I just mentioned 2 of them here. One is in Parkinson's disease. If you carry a loss of function in the GBA allele, your risk for developing Parkinson's disease increases dramatically. And the same thing is true for frontotemporal dementia, another adult onset disease, if you carry a haploid heterozygous loss of function mutation in the progranulin gene. So in Alzheimer's disease, same thing. So we have the allelic series of the ApoEs, which is associated with high versus low LDL receptor-mediated ApoE uptake. And consequences of that are lipofuscinosis in human neurons and the accumulation of pathogenic lipids in lysosomes. And recently, there is a study out that shows that heterozygous loss of function mutations in several lysosomal storage disease actually increase the risk for AD. And an interesting fact is also that this was shown for one of the NCL genes, and they crossed it into an Alzheimer mouse model. And if you cross in a heterozygous loss of allele of this CLN1 gene, you see that it greatly favors amyloidogenic phenotype. So the take-home message is, and this is widely accepted now, that the endolysosomal system is actually a genetic hotspot, right at the crossroad of childhood and adult onset neurodegenerative diseases. Now back to the metabolism, lysosome metabolism, and this is the great breakthrough in looking at lysosome metabolism and what goes wrong. Came in I think in 2022 when the group of Monta Abu Ramaili published this first paper. They had published the technology before, but this is really the first study where they showed that the technique of lyso-IP, where you rapidly immune precipitate lysosomes from tissues or cells, gives you a method to really study what goes wrong in the intralysosomal metabolism in different diseases. So Monta did this study for the CLN3. He built a CLN3 mouse model, and he also had cell models. And the characteristic of these models is that they all contain this the transgenic expression of a lysosomal transmembrane protein that is equipped with 3 H8 tags that allows you to rapidly immunoprecipitate lysosomes from live tissue or just rapidly isolate, freshly isolated tissue or cells. And these lysosomes are intact, so even the soluble metabolites remain, and you can analyze these by mass spec. And this is what he did and his group. And for CLN3, that way they found that CLN3 lysosomes accumulate degradation products of one particular lipid degradation pathway, namely the glycerophospholipids. So what he found is that the glycerophospholipids are degraded in several steps. So they have here you have the glycerolipids on the left here in this bubble. In the middle, they have after one breakdown step, one of the acyl chains is removed, and after a second breakdown step, the second acyl chain is removed, and you end up with molecules called glycerophosphodieesters. And CLN3 lysosomes accumulate massive amounts of these GPDs. And what they also accumulate are these intermediate forms, these so-called lysophospholipid forms, where a single acyl chain is still present. This, in the meantime, has been shown to occur in zebrafish models, in a mini swine model. These GPDs you can also monitor in plasma from CLN3 patients. And as Monter, working together with Andang Do at NIH, showed in the CSF of CLN3 patients. Now, very nice work was published recently because a study was done by Esther Zamler and her group, and working with different clinical centers. So they collected peripheral blood mononuclear cells, and they now used a so-called tagless lyso-IP technique, because of course you cannot tag it in patients with an H8 tag. So this is also based on an antibody, the Recus endogenous TMEM192. And what I found is that PBMCs, freshly isolated from patients, also accumulate massive amounts of these glycerophosphodieesters. And a very interesting finding in his first study is that in the so-called syndromic CLN3 patients, you see massive accumulations. But in this first cohort, they had also a non-syndromic CLN3 patient. And these are patients that have mutations in the CLN3 gene that are not as more than 80%, so the delta 7-8 mutation, but these are rare mutations that only give rise to a retinal phenotype and no neurological or very few neurological phenotypes later on in life, but they don't look like Batten. So the interesting thing is that in this non-syndromic patient, the levels are far, far down as compared to the syndromic patients. Now you can say, so what? So GPDs, yeah, so what? Lysosomes accumulate GPDs, but the lab of Monter and his colleagues in Stanford have also shown that these GPDs are not just innocent bystanders. So what they have shown is that these GPDs accumulate to levels that are far, far above IC50 to inhibit lysosomal lipases. So in other words, and this probably also explains why the intermediate degradation products accumulate. These enzymes that do this job here, they are just inhibited. Also, another important finding, and this is not necessarily coming from work from the group of Monter, but it was already published by other groups before, we know that in CLN3 cells, there's a lot of collateral damage. So if CLN3 is absent, what we see is that you also have it interferes with the catabolism, redistribution, and recycling of other lipids. One example is here on the right, cholesterol. CLN3 cells accumulate cholesterol. And so that's a bit like what one sees in Niemann-Pick disease, but it's not as obvious as in Niemann-Pick, but you clearly can see it. Another thing is what you see is that the sphingolipid breakdown pathway is also affected. And that is why you see accumulation of sphingolipids, such as the globoside GB3, which is shown here in the bottom of this histogram. And you see another ganglioside GM3, which also accumulates in CLN3 deficient cells. Here, there is one lipid I want to highlight, and for a reason that will become clear in a moment, and that is the lipid BMP. BMP is the rare lipid that is actually decreased, massively decreased in CLN3 deficient cells. And this was shown again first by Monter's lab using lyso-IP method. On the left, you see that in this plot, you see that the BMPs on the left, they're very much decreased, whereas the lysophospholipid, so lysophosphatidyl and choline are all increased. Now, what was also known earlier already from work from Susan Cotman's lab in stem cells, CLN3 deficient stem cell derived neurons, is that these cells lack multivesicular bodies. These are basically endolysomal compartments that carry interlysomal vesicles. And these vesicles actually contain BMP. And BMP, as I will show you in the next slide, is actually an incredibly important component of the lysosome because this lipid serves a key role in sustaining the activity and stability of lysosomal hydrolases. And it is here depicted here in this diagram with these red blobs. It provides a surface on which co-activators and lipid-related proteins, including the enzymes, the lipases, they come together and they can access their substrate. So when BMP is missing, there's a lot going wrong inside the lysosome. Now, at the beginning of my talk, I told you that at first glance, there seemed to be little or no overlap in the molecular pathways affected by loss of function of different NCL genes. Now, this is a view that has recently dramatically changed, and because of two remarkable discoveries and likely more to come. Now, the first one is shown here. So again, Monter, Aboramile, and colleagues have shown, using lyso-IP, that if you knock out CLN5, you actually completely prevent the formation of lysosomal BMP. And they discovered that this gene encodes the lysosomal BMP synthase. The second discovery came from a very different lab in the UK, from Kasparas Petkevicius, who was actually studying lipid remodeling and a group of enzymes involved in lipid remodeling. And in this group of enzymes, there's also CLN8. And he found that CLN8 actually functions as an extra lysosomal BMP synthase that converts RS-LPG into RS-BMP. And then the question is, well, the precursor for the intralysosomal SSBMP form is actually SSLPG. So the question mark here indicates that there's still something missing, something we don't know. Some people may know, but I have to say we don't know yet, is the fact that this RSBMP, which is outside the lysosome, has to be converted to SSLPG, which then has to be taken up in the lysosome before CLN5 can use it to make BMP. Another very surprising discovery recently, because many people thought that BMP is an extremely stable lipid, is undegradable in the lysosome, and etc. Now that dogma has recently been thrown out of the window, I should say, because it's now clear that there is a lysosomal BMP hydrolase, and this is called PLA2G15. And what is interesting is, so this is the one that is actually responsible for degrading BMP inside the lysosome. What is interesting is that two groups came together, namely the group from Monte Aberomili and a biotech company in Amsterdam called Scenic Biotech, who had studied Niemann-Pick and started looking at genetic modifiers of NPC phenotypes, and using a haploid cell model that lacks the Niemann-Pick type C1 gene. And they discovered that this molecule here, this PLA2G15, was a dramatic modifier of the NPC cellular phenotypes. They made knockout mice, crossed them with Niemann-Pick type C mouse model, and found that if you knock out this particular hydrolase, you dramatically increase the lifespan of the Niemann-Pick mice. So this finding seems to have very important potential therapeutic implications. But what it altogether sort of points to is that we now are more and more moving towards a model where we think that BMP dyshomeostasis is actually a very common hallmark in different NCL diseases. I mentioned that, of course, CLN5 and CLN8 are directly involved in the synthesis pathway. CLN3 has much lower BMP levels, but that's not only true for CLN3, it's also true for CLN11. So this is the gene that encodes progranulin. Also there you have very, very low BMP levels. So there seems to be a common pathway that is evolving, and I think in the next year or two we will see many more players fitting into this pathway. Now, coming back to CLN3 disease itself again, and some of the open questions. So I think what we have not answered today, although we have made great progress thanks to these technologies, etc., understanding maybe what all goes wrong, is why neurons die and what's the role of different brain cell types, astroglia, microglia, neurons, etc. So we have seen a number of things happening. We've seen the massive lysosomal accumulation of GPDs, and CLN3 being absolutely necessary for efflux of GPDs from the lysosome, such that they can be reused for maybe making new lipids, etc. We've seen the accumulation of potentially toxic lysoglycerophospholipids, because we know that many of these lysolipid forms have toxic effects in cells. We've seen the accumulation of other lipids, including gangliosides, GB3, cholesterol. We have impaired catabolism or efflux of lipid building blocks, and we have the accumulation of lipofuscin. I showed you the inhibition of lysosomal lipases by GPDs, the depletion of this important lipid BMP. What I didn't show you is there's also depletion of certain lysosomal hydrases that has been established through proteomics studies. I also didn't show you that there is mistrafficking of important receptors, such as the cation-independent M6 phosphate receptor, which is an important trafficking receptor for lysosomal enzymes. I'll come to defects in lysosomal repair. So the question in the end is, what is in the end causing neuronal death? Is it the reduced supply of building blocks, which has been shown by Monte's lab, but also by work from the Raimundo lab, a paper that is online. They showed that there are defects in the nuclear membrane, and with the consequence that there is DNA damage, the YAP pathway is activated, and this leads to pro-apoptotic signaling. So this could also be an important pathway that is involved in eventually the kiss of death for the neurons. We have seen glial dysfunction that's well known and said neuronal loss. An aspect that has recently popped up is, and I mentioned it already, what's the role of lysosomal damage in CLN3 cells? So is it maybe the integrity of the lysosome in the end that is the kiss of death for these cells? And a question that relates to that, would improving lysosome repair actually translate into therapeutic benefits? Now, Tan and Finkel have shown already a few years ago that if you knock out CLN3 in cells, you get a recruitment of a kinase called PR4K2A to the lysosome. And this is a pathway that is one of the key pathways that is involved in lysosomal repair. So this is clear evidence for that lysosomes are damaged, repair pathway is activated, and therefore the question whether this could be used to try and find new therapeutic agents by building assays, and this is work that is ongoing right now. I just wanted to say that the lysosomal damage pathway and lysosomal damage in general is now evolving in a variety of diseases as maybe a key aspect. It's been shown to occur in the context of LRRK2 Parkinson's disease. It's now been shown also to occur in the context of another NCL disease, CLN4 disease. And there is various papers describing lysosomal repair and repair defects in the context of Alzheimer's disease. And last but not least, talking a bit about the role of microglia in CLN3 disease. Here again, we all know that these are super important cells in the brain. They're the sort of the guardians of brain and retinal health. They are promising targets for therapy for two reasons. One is maybe if they play a dominant role, then impairing defects in microglia might actually have therapeutic benefit. Susan Cotton, in collaboration with Sabina Tahirovic, has shown that CLN3 microglia show lots of impairments. And they also have done some drug testing and shown that you can actually rescue some of these or normalize some of these defects. But I think an area that is much more forward-looking now is that, and also in CLN3 disease, given the critical role of microglia in CLN3 disease, targeted therapies that are based on brain-wide replacement of microglia with gene-corrected or genetically modified bone marrow-derived microglia-like cells might be a very promising avenue for therapy. And I've listed here below, without going into detail, a number of recent examples that have been published, which you can take a closer look at. Now, very briefly, today we are in a situation where most drugs that have been proposed to treat CLN3 disease, apart from antisense oligos, which I haven't gone into, or gene therapy, which I also haven't gone into, all the low molecular weight therapies and the hope one has from those, the effects have been based on using classical readouts, including accumulation of the subunit C, GB3 accumulation, improving autophagy, looking at lysosomal parameters, etc., etc. Bottom line is that today we are still in a situation that we do not know what all these drugs really are doing at the omics level when we analyze the lysosomal metabolome. And this is work that is currently ongoing. So what we hope to get at is actually a much clearer picture of what these drugs can do and what they cannot do. And with that, we are actually hoping that these novel insights that we have gained recently and these novel technological approaches, including the translation from the animal model to human PBMCs and measuring molecules that are certainly promising, maybe as biomarkers, that we will reach soon an inflection point and can move actually from symptomatic to disease transforming treatments. And I think with that, I come to an end, and I would highly encourage you, if you have specific questions, my email addresses are on the title slide, and I'm very happy also to have online meetings with anybody who is interested in learning more or having a more detailed insight in some of the things I basically showed you in a helicopter view. Thank you very much.